recent developments. What are the recent developments and how do they affect the biosecurity at the poultry farm? How you have to update your biosecurity, and a very practical and important aspect that I'm going to speak about. And I'd like you to have your good attention so that whatever I'm telling you, you can apply it at your farm. Wherever there is poultry, there will be diseases. In spite of our best efforts, it's not possible to prevent them. They come, produce disease, make birds sick, reduce production, and even inflict heavy mortality. We try our level best at the farm to prevent these diseases, but even then, they keep coming and take a heavy toll. Why? Why happened this way? And what can we do about it? That's going to be the thrust, that is going to be the focus of my talk. I want you to have a good look at this triangle. On the top is disease, then on the right, vaccination and immunity, and here is biosecurity. In my talk will revolve around these three aspects. I will start from disease, then go to vaccination and immunity, and then at the end, I will come to biosecurity as to what we can do about biosecurity. First, let us see what the disease is. As we know, that disease is produced in poultry by bacteria, by viruses, by fungus, by mycoplasma, and many other organisms. How powerful these enemies are, these disease-producing organisms, how powerful they are, how strong they are, the enemy of birds. Let's have a quick look about them. Now, here are some facts. Very quickly, if bacteria, they multiply within 50 minutes, from 1 they become 2, what is known as the doubling point. How bacteria multiply? From 1 to 2, 2 to 4, 4 to 8, 8 to 16, and like that. It is called binary fission. Now, if this is the standard, this is standard. If the doubling time is 50 minutes, then just within 24 hours, they become 500 millions. That means within 24 hours, they become 50 crores. Now let me give you an example. E. coli, doubling time is only 20 minutes, from 1 to 2 within 20 minutes. And if you calculate it, within 9.5 hours, from 1 E. coli in the bird when he enters, it becomes 50 crores. See the amazing, the power, their reproductive capacity, how fast they multiply. Let me give you one more example. E. coli, of course, is the very most important bacterium of poultry. Very common, extremely common. Every farmer, every person who is dealing with the poultry knows about what E. coli is. Then another example is what is the third one, Clostridium perfringens. Clostridium perfringens, its doubling time is only 7 minutes. Its doubling time is 7 minutes. Within 3.5 hours, this Clostridium perfringens divides into 50 crores. This Clostridium perfringens, friends, you know, it is an organism which is responsible for necrotic enteritis. Necrotic enteritis is a very common disease, particularly of broilers. And you find that this bacterium, it suddenly attacks, multiplies at a fantastic rate, and within few hours produces the disease and inflicts heavy mortality, Clostridium perfringens. So bacteria, they can be very powerful and their reproductive capacity is very high. Now let us examine viruses, because most of the diseases produced in birds are caused by viruses, like Ranicate disease or Newcastle disease, avian influenza, infectious bronchitis, infectious laryngotracheitis, so many diseases, adenoviruses and all that. Viruses, what happens? Let us try to see. Viruses are even more powerful than bacteria. How? Supposing this is my fist. This is a body cell of the body. This is body cell. Virus comes. For entering within this cell, it has first to attach itself. The virus, let us say avian influenza virus, it has entered the bird through the respiratory system. When it goes into the respiratory system, the first thing it does, it attaches itself to the lining cells of the respiratory tract. After attachment, it enters within the cell. So it enters inside this cell. After entering inside this cell, it multiplies at a very rapid rate. So much so that within few hours, within 3, 4, 5 hours, it becomes not only in billions, but it multiplies into trillions, billions and trillions, not only in millions like bacteria. So in millions, billions and trillions, they accumulate within the cell. How they are able to do that? This is because after viruses enter within the cell, this is a basic difference between a bacterial infection and a viral infection. After the virus enters within the cell, it takes complete charge. It takes complete control of the cell. And cell is therefore not able to do its own activity. On the other hand, cell becomes a factory for the manufacture of viral proteins, viral particles. The virus takes a complete control of the cell, and it becomes virtually a factory for the manufacturing of the virus. That's how they, within short period, multiply in billions and trillions. Then the cell bursts. Viruses are liberated. They infect the other cell, and that is how they destroy the cells and the organ, and the bird dies. In bacterial infection, the story is different. Bacteria are outside the cells in the bird's body. They don't go inside. And they kill the bacteria, E. coli, perfringens, they all kill the birds through toxins. So there is a basic difference. You should try to understand this basic difference, because in my subsequent discussion, I will give a reference to these. So the basic difference is viruses multiply within these cells, bacteria outside, and viruses, they take full control of the cell and multiply. And that's why it is difficult to control them with or treat them with antibiotics. Antibiotics do not enter the cell. Antibodies do not enter the cell, so viruses are difficult to control. With that brief introduction, the point I was trying to emphasize is, let's go back to this one. The point I was trying to emphasize is, this much about disease. That disease is caused by our viruses and pathogen. They are very strong and powerful enemy of the birds. Very strong and powerful. Within few hours, they can kill the birds. Classical example is avian influenza virus. Within 48 hours, avian influenza virus, highly pathogenic virus, can inflict up to 100% mortality in the flock. Every bird dies. How powerful they are. Okay. Now, I come to the second part of the story, vaccination and immunity. Now, we try to prevent the diseases at the farm by these two basic approaches. One is vaccination. We carry out vaccination to produce a very strong immunity so that the birds remain protected. And we also exercise good biosecurity so that the birds, the disease-producing organisms, I call them as pathogens, they do not come from outside into the farm. By biosecurity, we try to prevent the entry of disease-producing organisms into the farm. So these are the two approaches we are using. But in spite of these two approaches, in spite of the good vaccinations that you carry out, the best vaccines that are available to you, you carry out vaccinations properly, you follow all the instructions given by the manufacturer, best vaccine, best procedure and everything, and yet diseases keep coming. They inflict mortality, they inflict heavy losses, and they affect your profitability, your profits and all that. Why? Now, this is the aspect I will be briefly discussing, and then I will at the end come to the biosecurity as to what we can do about biosecurity. Now, imagine a situation. Supposing again I give the example of my fist, and take this fist as a virus. You take this fist as a virus. This virus enters into the bird. The bird's immune system produces antibodies to kill this bird. To kill this virus. That's why they're called as neutralizing antibody. Avian influenza, for example, when the infection occurs in avian influenza or any viral disease for that matter, the bird produces antibodies. These antibodies are neutralizing antibodies. What they do is they try to go and they attach to the virus and they prevent their entry into other cells. They neutralize its action. They try to destroy the virus. So supposing you have carried out a vaccination against one particular disease, let us say avian influenza. This is because in the meantime, the proteins on the surface of this virus, they have changed. And when the proteins have changed, the antibodies which were produced earlier, they are not able to recognize this virus now. This virus has altered itself, and that's why they are not able to recognize. There are no homologous matching. There is no proper matching, and therefore the antibodies are not able to destroy this virus anymore. If you have followed this basic principle, this is what is exactly happening in our poultry today. And I will give you 3 very important examples to illustrate my point. Number 1 is avian influenza I will talk about. The second disease I will talk about is infectious bronchitis. And the third example I will give you is that of mycoplasmosis. Now let us first take avian influenza virus. Friends, these viruses, whether it is avian influenza virus or infectious bronchitis virus or mycoplasma, Mela disease, whatever they are very strong enemy of the birds. They are very powerful. They have an amazing capacity, reproductive capacity we have talked about. They multiply in millions and trillions within few hours. They become resistant to disinfections. So it is very difficult to control them. Now, let us see what is happening under field conditions. Let us take an example of avian influenza. I will not go into technical details. I will only touch those basic points which concern us from a field point of view. This is the virus of avian influenza. In the avian influenza virus, this black circle, this is what is called as an envelope. They are RNA viruses. They have an envelope which is composed of lipid. On the envelope, there are 2 projections. You can notice that. Upper red one is called as hemagglutinin on the top. That's right, this is the one. And there are these 16 hemagglutinins in the avian influenza virus. And the yellow one is what is called as neuraminidase. And there are 9 neuraminidases, which means 16 multiplied by 9 is equal to 144. This avian influenza virus, at least theoretically, can exist in 144 different combinations under field. Now, supposing there is a situation, avian influenza is an extremely common disease. The highly pathogenic, highly harmful, most harmful form, highly pathogenic, is under control. But the low pathogenic avian influenza virus has become endemic. It has become a permanent resident of our country. Throughout the country, it is there. And it has come to stay here permanently. We have to understand this enemy very well. If we do not understand this enemy very well, we will not be able to plan any good strategies for its control. We must know what are its strong points, what are its weak points, and what we can do about it. Now what happens is that supposing in the field, here or elsewhere in India, the field virus is H9N2. And the vaccine that you are using is containing it H7N2. There are 144 different combinations, I told you, and most of them have been reported from the world. If the field virus is H9N2, and if you are using the vaccine of H7N2, there will be no protection. Your vaccine will go waste. It cannot give any protection. Why? Because there is no cross immunity between the subtypes. Subtype is H7N2 is a subtype. H7N2 is a subtype. Within subtypes, there is no cross protection. So, if it is H9, the point was this, without making those complicated words, the point is, whatever field virus is there, if your vaccine contains the same virus, then only it will give the protection. So what can be done about it? What are they doing abroad? In United States, in the Western world, in China and other countries, what they are doing is, they are regularly monitoring the type of the virus under field conditions. They have very good laboratory, they have got very good experts, and they keep monitoring throughout the year the field virus and produce their vaccine accordingly. They monitor the virus and update their vaccines all the time, which is not happening in our country. We are not able to do that. So this is one thing. Now, let us come to the other aspect of this virus. Now, the top one, hemagglutinin, the top one, and you put it on the top. Yeah, because of its high rate of multiplication, if it commits mistake, one should combine with two, but it does not combine with two, one combines with three. If one combines with three, that is an error. That is a mistake. And this mistake is nothing but a mutation. Mutation in Latin means a change. There is a change. Wrong base pairing. Because of that mutation, where does this mutation occur? It occurs in the gene. See, in the center, there are 8 segments. You can see 1, 2, 3, 4, 5, 6, 7, 8. These are RNA, and these 8 segments are the genes of avian influenza virus. What is the role of this gene? Supposing that central gene, this is the one which is responsible for encoding hemagglutinin. Friends, just simple, in a simple way, whatever proteins are formed, not only in the virus or bacteria, but also in our body, the instructions which protein, which amino acid is going to be formed, they come from the gene. It's a very complicated mechanism. So gene says you produce the normal amino acids in hemagglutinin. But because of mutation, it produces another amino acid. So what happens? On the tip, there is a change in the amino acid composition, in the sequence of amino acids. And because of this change, the antigenic structure of the virus is changed. And therefore, the antibodies which were formed earlier, they are not able to work. They are not able to work. So much about avian influenza virus. I will not go into any details. Now I come to the second example, that of infectious bronchitis. Very important disease, very common disease, as Dr. Reddy was telling the other day, throughout India, not only throughout India, throughout the world. Throughout the world. The virus of infectious bronchitis was discovered in the year 1930. That means nearly 78 years ago, more than 70 years ago. Still we are struggling with the disease. We have not been able to form the proper vaccine. We are not able to control it. Why? This is because this virus is not stable like avian influenza virus. It is an RNA virus. It undergoes mutations continuously and changes itself continuously like avian influenza virus. Now, one more important thing, I will not go into the details, that nature has given a mechanism in viruses that if it makes a wrong combination 1 combines with 3, it is able to correct itself. This mechanism is present in DNA viruses, but this mechanism is not present in avian influenza virus. It is not present in infectious bronchitis virus. And therefore, the mutation which occurs, it persists. It is a permanent change. It is an irreversible change. Okay, so infectious bronchitis virus, infectious bronchitis. Infectious bronchitis is a linear virus. It also has a very high rate of mutation. It changes its surface proteins all the time, and when they change, the earlier formed antibodies are not able to work. So you are required to give another vaccine, and this story goes on and on and on, and we are not getting anywhere. One more interesting thing about this virus, in case of avian influenza virus, it changes only through mutations. This virus also changes through mutations, but there is another one strategy it adopts, and that is what is called a genetic mutation, not genetic recombination. Let me explain to you in a simple way what is genetic recombination. What happens is there are different serotypes of this virus because of the change. Now, supposing there is an infection in a bird of IB virus, infectious bronchitis virus, and 2 viruses are present in the same cell of the respiratory tract. They are present in the same cell of the respiratory tract. When they come in close contact with each other, they exchange their nucleic acid. And this is what is called as genome recombination or genetic recombination. I have shown it there. On the top, you find the virus. There are 2 serotypes which are there. Then, in the second, there is a cross. There is an exchange going on. This is IB virus. And here in the third, you find that red portion has come here, that one. That small segment of red. And here the small segment of, in the long one, black. A small portion of the nucleotides have changed, and because they have exchanged, they have formed new serotypes. And this process is continuously happening with IB virus under natural conditions. It is a natural phenomenon. It does not stay stationary. It keeps changing all the time. Now, we face a situation. You have a standard vaccine. You use it at your farm for IB with the hope that by using that vaccination, it will produce 100% protection. It will give good immunity. But it doesn't. Why? Because the virus is changed virus. You had given in the vaccine, which was a different virus. The field virus, which is preventing and making the bird sick, is a different virus. Different serotypes. And there are not only 2, 4, 5, 6, there are nearly 100 serotypes. It is not possible to include all those serotypes in a vaccine. They are called as antigenic variants. Antigenic means they have changed their protein and they have a different form, variants. So this is what is happening continuously with IB virus, and we are not able to control it. And now I come to the last example, and that is mycoplasma. This portion on the top, that one, this is the lining epithelial cell of the respiratory tract. It's a lining cell of the respiratory tract. And these flask-shaped organisms, they are mycoplasma. And they have got what is called that black portion on the top, this one. It is called the attachment organelle. This is an electron microscope. You can't see with the ordinary microscope, electron microscope. And with this attachment organelle, they attach to the living cells of the lining respiratory tract. After attachment, they enter inside this cell and produce disease. Now with this virus, 2 things are happening. With this mycoplasma, 2 things are happening. One thing is the same thing which we find with avian influenza virus and IB virus. What same thing? It undergoes mutations in the environment. In the environment, it continuously undergoes mutation. This is one thing. IB virus was also undergoing mutations in the environment. But IB virus devised another strategy also of genome recombination. Mycoplasma is still a step further. It has come out with a novel strategy. What it does it, after infecting the bird and entering within this cell, when the antibodies are produced against it, and when the mycoplasma feels threatened that these antibodies are going to kill me, then within the bird itself, it changes its surface antigens, the surface proteins, it changes within the bird itself. It's not mutation. Scientists are working on it. It's called as a phase variation. And the mechanisms involved are epigenetic mechanisms. I will not go into those things. A lot of research work is going on. Hundreds of scientists are after these, how to prevent these, what is called as on and off switching. What happens supposing I don't want this light? I can switch it off. If I want this light, I can switch it on. The same thing is happening here. Mycoplasma has large number of genes, and they switch on and switch out and switch on and off those genes. What for? To change the surface proteins so that they are not killed by the antibodies. Friends, it's like something, supposing there was a fancy competition in the school, a fancy dress competition. There was a college day, and there was a fancy dress competition. One boy was asked to perform or portray the role of a beggar. He came with a fantastic makeup. Absolutely. I mean, nobody could make him out that this man, everybody thought he is a beggar. And he portrayed his role on the stage. Such an effective impact it had that even his friends could not recognize him. Then it so happened that boy's parents had also come to see this program. And the concerned teacher, he asked the parents, could you recognize that boy? And parents said, no, we could not. And the teacher told to their parents, his parents, he was your son. And the parents could not recognize his own son because he had changed his morphology, his huliya badal di thi, he changed his appearance. Exactly the same thing is happening with mycoplasma. When mycoplasma enters the bird and when it feels threatened, it changes its surface antigens within the bird. What is called as on and off switching. These are the recent developments. It's in my topic heading, recent development. These are the recent developments. When it changes its morphology, vaccines become ineffective. Not to that extent they are not effective. Antibodies are not able to kill it properly. What are you going to do? Very difficult to control. What are we going to do in that case? That's the last portion of my talk, and now here I come to biosecurity. I will not go into those things. Phenotypic on and off switching, immune evasion. Mycoplasma gallisepticum has an inherent mechanism. Inherent means it's genetically there for making rapid and sudden changes in the expression of its surface proteins in response to antibodies. Scientists have worked out, I will not go into those things, as to which are the genes which are switching off, which are switching on and off, and which are proteins they are changing. Still a lot of work is going on, but this is not our subject, we will not go into that. Now, this is the last but one slide. We have carried out, supposing you carry out, I focus now on biosecurity. Whatever I have discussed so far, the basic technical information, now we are putting it to application. In your farm, you apply this knowledge. Here you find that immunity you are carrying out, vaccination, the first one here on the left top, here is one. Here, about 80 to 90% immunity is there. You have carried out a vaccination and you find a very good immunity. So even if the biosecurity is poor, it doesn't matter. The immunity, strong immunity will protect the bird. Can you figure out a disease in which such a thing can happen? Where would be a disease in which you may get 80 to 90% immunity? And if your biosecurity is weak, immunity will take care of it. Can you tell me, any one of you would like to give me an example? Well, I will give, the example is Ranicate disease, Newcastle disease. It gives a very strong and powerful immunity. So even if the biosecurity is little weak, no problem. It takes care. Now, why Ranicate disease virus, a Newcastle disease virus, is giving 80 to 90% immunity? On the other hand, if we go, there are various changes, and if we come to this last one, we find that there is only 50% immunity. Only 50% immunity. Now, why this kind of a picture is there? In case of the first one, in case of Ranicate disease, why there is good immunity, here very poor immunity. The reason is Ranicate virus is stable. It does not undergo mutations like avian influenza virus, like infectious bronchitis virus, or like mycoplasma virus, or even like Marek's disease virus. It is stable. Throughout the world, there is only one serotype of virus, Ranicate disease virus. What does it mean? It means if you are using the vaccine here in Kolkata, the same vaccine will be effective in Chennai, the same will be effective in United States, in Europe, in China. One vaccine. Why? Because the virus is stable. So you have an effective vaccine and good immunity. There are 18 genotypes. The latest is that there are 18 genotypes. But these genotypes are not affecting because it doesn't change the protein. Protein is changed by mutation. And this Ranicate disease virus is stable. Now, in the subsequent story, there are various degrees of vaccination percentage. Sometimes you may get 60, 75, and here you may get only 50. Now we come to the last slide. Why 50 alone? Classical example is infectious bronchitis virus. Because the virus is changing all the time. There are so many serotypes, and the vaccine that we give may not produce the adequate immunity. It may produce only 50% immunity. So what can we do about these things? If there is only 50% immunity, vaccines, let me make it very clear, don't carry a wrong impression. There is nothing wrong with the vaccine. Vaccines are good. You are following all the procedure to implement your vaccination. But even then they are not giving full protection for the simple reason that the virus is changing all the time, in avian influenza, in infectious bronchitis, in mycoplasma and all that, even in Marek's disease. And that's why the vaccine is not able to cope with the situation. This is the limitation which farmers must realize. You should realize this thing, that if you are under the impression that you have carried out the vaccination and your birds are fully protected against infectious bronchitis or avian influenza or mycoplasma or Marek's disease, that's not the story. You are under a wrong impression. Now, if the vaccine is giving only 50% more protection, what are you going to do? Who will come to our rescue? Who will come to the rescue? The rescuer will be biosecurity. If you get 50% protection with the vaccine, you strengthen your biosecurity to such an extent that you compensate with the deficiency of immunity with strengthening your biosecurity. Make your biosecurity more strong, effective, powerful. The purpose of my talk given to you is that you are helpless. You are using the best possible vaccine. You are doing the best possible biosecurity. You are doing everything. But even then, you are not getting good protection. Why? Because of the problems associated with the pathogen. They are changing all the time. If there are limitations of the vaccine, there are limitations with the vaccine, you cannot get full protection, then to your rescue will come the biosecurity. You strengthen your biosecurity. And how you strengthen biosecurity, I will give you a couple of examples. Now, we have talked about infectious bronchitis virus. Okay, I asked you a question. All of you are familiar poultry farming. Supposing you have a farm, poultry farm here. good population, let us say 5,000 birds or 10,000 birds. And there is another farm very close to your farm with 10,000 birds. Not even a half kilometer away, even less than half a kilometer away. Now that farm is having infectious bronchitis infection rampant. There is a very high level of infectious bronchitis virus infection prevailing on that farm. Your farm is also very close. Now my question is, how many of you would think that virus, because his farm is having IB virus, by air, it can come to your farm and infect your birds? From the neighboring farm, the virus through air, you are afraid all the time that my farm is surrounded by vicinity with those farms where the diseases are prevalent. Let us take an example of IB virus. How many of you will think that that IB virus will travel by air, come to your farm and infect your birds? Please, give what you think. Will it come and infect the birds? It will come and infect the birds. How many of you feel that it will come and infect the birds? Everybody? This is the wrong notion. This is the wrong impression. I have talked about the strong points of viruses and bacteria, how strong they are, about their reproductive capacity, about their breeding capacity, about their disease producing capacity. Now let us have a look at their weak points also, where we can hit and make our biosecurity more effective. They cannot come from that distance. Why? If you go through the literature, the facts are that IB virus travels from one pen to another pen. At the most, it can travel from one shed to another shed at your farm only. Supposing your farm is having 10 sheds, then it can spread within sheds, but it cannot travel long distances. Why? It cannot travel long distances. Try to understand this thing, because nobody will be able to tell you these things. These are the new developments. It cannot travel long distances. It is an envelope virus. Envelope is of lipid, and when it travels more than a furlong or something, it gets inactivated. It gets killed in the in the environment. This is the truth about it. Now, when it gets killed in your farm, if you practice a very high quality of biosecurity, the virus is not going to from outside. Supposing if you are doing regular spraying, people spray with disinfectants at the farm without belief that this is a ritual, it's a routine practice, it's not going to work. It will work. It will destroy these viruses. Whatever are present in your farm, those viruses will be destroyed. And no virus is going to come from outside. Then who will this virus to your farm? People coming from outside in their shoes, vehicles and all that, and you know how to tighten your biosecurity. Now, the source of this infection are the birds themselves. How? Supposing the birds have suffered from IB infection at your farm. They have recovered. Supposing some may have died because of secondary complications with bacteria like E. coli, and others have survived. Those birds which have survived IB infection at your farm will harbor this virus, will keep this virus latent form in cecal tonsils and kidney, not in respiratory tract. That also is a new knowledge which is coming up, that they are stored in kidney and cecal tonsils. And when there is a stress, the virus gets activated. So if you exercise a very rigid, very strict, very rigorous biosecurity at your farm, it will solve a lot of problems. And that's where I come to the last slide. This is my last slide, conclusion. I have given a formula: excellent biosecurity, immunity, excellent management is equal to excellent protection. Now let us examine this formula. What does it mean actually? I have given excellent biosecurity and excellent management But before immunity, I have not used the word excellent. You could notice that. I have said excellent biosecurity, simple immunity, and excellent management. I have not written excellent immunity. We wanted excellent immunity, but I have not written. Can any one of you try to explain why I have not written excellent there? Can you figure it out? We all want excellent immunity. Every farmer wants excellent immunity against that particular disease after carrying out vaccination. But you are not sure what percentage of immunity you are going to get because it is not in your hand. Biosecurity is in your hand. So if you work very hard, you can produce excellent biosecurity. If you can work very hard, you can have an excellent management because those two factors are in your hand. Immunity, what percentage immunity you are going to get is not in your hand. You sincerely use good vaccine, you carry it out, follow all the procedures, then it depends on nature as to how much immunity you are going to get. Because it's a natural phenomenon. Virus keeps changing all the time. You cannot prevent it. You cannot say that avian influenza virus is stop doing this mutations business. It's creating a problem for me. IV virus don't undergo mutations. Mycoplasma don't use all those strategies. We are facing problem. But it's not in our hand. Because these are the natural phenomena. They will go in nature. You can't do anything about it. We simply have to understand them. That's what the scientists have worked out. What are their strong points and what are their weak points. So you improve your biosecurity. Another example I can give you. How many of you will feel that there is a farm here, there is another farm. This farm is having mycoplasma. It can come to your farm by air. Mycoplasma is a very common infection, very important, predisposes to many diseases. If it is in a neighboring farm, can it come to your farm through air and infect your birds? When there was a first outbreak of avian influenza in our country declared by the government of India 2006 or something, I was invited to give a talk. And I said, if avian influenza, highly pathogenic virus, if it is present in West Bengal, I come from Madhya Pradesh, can it come by air to Madhya Pradesh? Farmers were threatened at that time. They were scared. They were scared stiff that virus may come by air and attack their birds. And everybody raised their hand, yes, we are afraid that the virus may come from West Bengal and it can come to Madhya Pradesh. I said, no. Americans have done a lot of work on this virus. It cannot travel more than 45 meters, not kilometers, sorry, meters. That means very strong, very nearby. It cannot travel long distances. Mycoplasma cannot travel long distances. Mycoplasma from one pen to another it can go. Even it does not travel to sheds also. They are very delicate. They are easily destroyed by disinfectants. All these enveloped viruses, friends. Raniket disease virus is enveloped. Infectious bronchitis virus is having an envelope. Avian influenza virus is an envelope. All the enveloped viruses, they are easily destroyed by disinfectants. They are very fragile. They are delicate. Most people have a wrong impression that envelope is there, so it will protect the virus. It does not protect. Why? Because that envelope consists of lipid, fat, and the disinfectants easily dissolve the fat and kill the virus. They do not survive for 3 to 4 days outside. So the point I'm trying to emphasize is, if you carry out very strong immunity, then it can come to our rescue, because we will get only limited protection with vaccines. In Raniket we get very good protection, but not in many other diseases, because they keep changing all the time, and which is not in our hand. So this is the last thing, that in immunity I have not put excellent, because it's not in our hand. It's a natural phenomenon. What is in your hand is to use the best vaccine available. What is in your hand is to follow the manufacturer's precautions. But even in spite of all the precautions and the best vaccine, what percentage of immunity you are going to get, you are not sure. That's where we must strengthen. We must further strengthen whatever biosecurity you are exercising at present in your farm. You advise the farmers to strengthen it further. That will come to your rescue. The deficiency or the shortcoming of immunity from using vaccine will be compensated by very strong biosecurity. Friends, thank you so much for your attention and for your patient hearing.